Welcome to the Explainer. Today, we're diving into one of the most astonishingly complex and dynamic systems known to science. Seriously, think of the adaptive immune system not just as a set of biological bodyguards, but as this massive, ever-evolving library of defense mechanisms. Every single day, your body is referencing this library to identify and neutralize foreign invaders. But how do researchers actually go about reading these billions of unique books? Well, thanks to some massive leaps in sequencing technology, we can now decode this ultimate defense library with unprecedented clarity. All right, here is our roadmap for today. We'll be moving through 5 key areas. First, the immune defense library, then scanning the repertoire, followed by the 6-step workflow, frontiers in immunology research, and finally, we'll wrap up with CD genomics sequencing solutions. Let's get right to it. Section 1, the immune defense library. Let's start by understanding the body's catalog of immunity. So, what exactly is the immune repertoire? Simply put, it's the complete functional catalog of your T and B cells. Operating independently but complementarily, these cells are constantly circulating through the system, basically on standby for their specific targets. When we measure the diversity of these cells, we're actually measuring the overall immunological complexity and readiness of the entire defense network. It's the ultimate biological readiness test. Now, the sheer scale of this library is, well, it's literally astronomical. These T-cell and B-cell receptors, TCRs and BCRs, are like highly unique molecular keys capable of recognizing an almost infinite number of foreign invaders and internal host factors. And the secret to this astronomical diversity, it all comes down to a sequence called the complementary determining region 3, or CDR3 for short. Created through highly dynamic genetic recombination, the CDR3 region is the absolute epicenter of variability. It dictates exactly which specific lock a particular immune cell can open. Moving right along to Section 2, scanning the repertoire. How we went from reading single books to scanning the entire library. You know, in the past, studying this repertoire relied heavily on traditional strategies. Think spectroscopy typing or Sanger sequencing. You were essentially trying to read a library of millions of books, just one single letter at a time. It was incredibly laborious, it was costly, and to be honest, it was pretty low resolution. But next-generation sequencing, or NGS, completely flipped the script. It's a real game changer. It provides an unprecedented, high-resolution snapshot, letting researchers read millions of those books simultaneously. That high-throughput capacity generates enormous data, allowing us to track dynamic changes across the entire cell population. Okay, Section 3, the 6-step workflow. Let's trace the technological journey of a biological sample. Turning a physical sample into actionable data follows a surprisingly crisp 6-step workflow. First things first, cell isolation. We grab the specific B or T cells we need. Next up, template purification and amplification. We target those highly variable CDR3 regions we just talked about. Think of this as selectively photocopying only the most important pages of our massive library. Step 3 is constructing the actual library and running the high-throughput sequencing. Once we have our millions of reads, Step 4 kicks in to filter out the noise. We want clean data. In Step 5, those cleaned-up sequences are aligned and clustered back to known gene segments. And finally, Step 6 is where the magic happens. Deep data analysis to accurately quantify the diversity and clonality of the immune system. Let's dive into Section 4, Frontiers in Immunology Research. This is how we translate all that data into massive biological discoveries. Next-generation sequencing of the immune repertoire unlocks absolutely incredible applications, specifically in fundamental basic research. By mapping out exactly how immune cells respond to specific stimuli, researchers are rapidly accelerating the discovery of novel biological indicators. It lets scientists decode the really deep underlying mechanisms of how the immune repertoire actually develops over an organism's lifespan. And even more fascinating, it helps us understand how the immune system interacts with the vastly complex microbiome living right within the host. To highlight just how powerful this is, let's look closer at those host microbiome interactions. Researchers have actually discovered circulating T cells that are uniquely reactive to the friendly bacteria living in our own gut. That is wild. It basically proves our gut bacteria are actively shaping our immune library. They heavily influence T cell cross-reactivity, which means your microbiome is essentially acting as a personal trainer for your immune system, constantly prepping it to respond to a much wider array of foreign antigens. Finally, Section 5, CD genomics sequencing solutions. Let's look at your partner in immune repertoire exploration. So, here is the million-dollar question for any active scientist watching this. If you want to leverage these high-resolution discoveries for your own basic research, how do you actually do it without having to build a dedicated NGS facility and hire a whole team of bioinformaticians from scratch? Well, CD Genomics provides the answer with some seriously tailored end-to-end sequencing solutions. Take that crucial amplification step we mentioned earlier, for example. CD Genomics gives you options. You've got multiplex PCR on one hand and 5-RACE on the other. Multiplex PCR targets the CDR3 region. It's a simpler operation that yields highly complete data, but, it can introduce some PCR bias. It's kind of like accidentally printing one page of our library 100 times and another page only once. Now, 5-RACE, on the other hand, targets CDR1 through 3. It's more laborious, for sure, and you might lose some partial sequences, but it brilliantly minimizes that amplification bias, giving you a much truer picture of the repertoire. The beauty is you get to choose the exact technique that fits the unique needs of your specific project. Partnering with CD Genomics provides way more than just raw sequencing. We're talking about a comprehensive one-stop service, the whole shebang, covering everything from initial experimental design all the way to advanced data mining. They utilize those cutting-edge amplification techniques we just compared, and they pair them with enhanced error correction algorithms. These algorithms actually use multi-layer clustering to fix sequencing errors on the fly. This ensures incredibly high reproducibility and clone consistency. They even have ways to align low-quality reads with high-quality clones just to recover important data, making absolutely sure you get the most accurate clonal quantification possible from your valuable samples. Now, I want to emphasize something absolutely crucial about the core focus here. As explicitly noted in their guidelines, the powerful sequencing solutions provided by CD Genomics are dedicated exclusively to advancing fundamental biological research. The goal here is uncovering the basic mechanisms of life in the laboratory setting. It's purely about empowering scientists to decode the biological truths of the immune system. So we've seen how the immune system functions as this vast, incredible library. We've seen how next-generation sequencing gives us the precise tools to actually read it, and how CD Genomics provides the specialized service to make it all happen seamlessly. With the ultimate defense library now wide open for high-resolution scanning, the only question left for us today is, what groundbreaking discoveries will your research uncover next? Thank you so much for joining me on this journey through the immune repertoire. Keep exploring, keep questioning, and keep learning.